Great. Well, thank you very much for that introduction, Josh. When I got the email inviting me to come speak at the meeting, I immediately responded yes, partly because I can think of only one thing more fun than talking about sex all day. But secondly, because I really literally grew up in the shadow of Warshoe, right off Skinker Boulevard. Used to have softball practice on the fields at the undergraduate campus, and so it's really such a delight and an honor to come to this great institution now in my professional capacity. So I've been studying the sex differences in the brain for, I guess, about 25 years now, and it has continued to surprise me in the things that are actually mediating these sex, trying to get at the mechanisms mediating sex differences. Okay, why are we not moving? I don't know why that's. Uh-oh. Oh, we were doing so well. Okay, hold still, let's start again. All right, now why is it, do I need to use the clicker to advance? That would seem strange. Right, exactly. That would not seem to be the smart. One thing is this, it's not sealing well. Let's try again. Okay, don't touch anything. Aha, okay. So sex differences are determined, I can go through this quickly, genes, environment, hormones, we all know that. And animal models are the best way to explore the biological basis of sex differences because we can do away with all of that cultural and societal influences. My animal model is the rat, and that's because it's not nearly as strain dependent as the mouse, and the sex differences are robust and reliable. So sexual differentiation of the brain, again, Art has already gone through a lot of this, so I'll go fairly quickly. But just to remind you all of the basic Bio 101, you start out with a bipotential gonad, and it is by default going to develop into an ovary. That is unless you have a Y chromosome with an SRY gene, in which case you're going to divert away from the default and form a testis. Now, what you might not always think about is actually that the brain is also bipotential. Well, first let's talk that testis, its sole job, as we've been hearing, is make sperm and make hormones, make testosterone being aromatized to estradiol. The brain is also, though, in terms of its phenotype, its sex phenotype, bipotential, and it will, again, by default, develop along the female phenotype. And you can think of this as that evolution devised it so that brain sex would match gonadal sex. Evolution doesn't really care about your chromosomal sex, despite Art telling us everything goes back to the chromosomes. From an evolutionary point of view, we want to make sure your behavior and your gonads match up in order to get to that next generation and reproduce. So your brain's going to become female unless you had that testis, which is going to make the estradiol, and it's going to convert the brain away from masculinization and convert it to masculinization. And in my world, we can do this as a readout to look at sex behavior as a measure of the masculinization of the brain versus feminization. Again, you can sort of think of it, the default female, God intended us all to be female, the devil came along, testosterone. Okay, so this is a great model of sex differentiation, but we now know that just about any endpoint you can think of is modulated by sex, and we've been making that point all morning. But when we think about the panoply of things that happen when the brain develops, starting with neurulation, I will say I don't think anybody has looked at the actual neural tube for sex differences, but very early on, neurogenesis, gliogenesis, neuronal migration, dendritic growth and myelination, synaptogenesis, synaptic pruning, and apoptosis are all influenced in some part of the brain at some time by gonadal steroids. And we could spend the whole rest of the day going through examples of all of these amazing effects on brain development. But instead, what I want to do is interrogate one system in great detail. And it was mentioned today some of the brain regions that are different, but it left out which is really the brain region that has the greatest sexual dimorphism, which is the preoptic area. Has nothing to do with vision, it's just named for the fact that it's it's rostral to the optic chiasm. But it is sort of the central processing unit for male sex behavior. If you lesion this brain region, males lose all interest in sex. If you stimulate this brain region, males lose interest in anything but sex. So it is an essential area. So it's a great model system to look at the mechanisms by which sex differences are established. And what we've been doing in my lab is looking at very specifically at spinogenesis, okay, the formation of dendritic spines, which you all know is the place of formation of excitatory synapses. And this is just a nice color coding of somebody else's work on dendritic spines, but these little mushroom-bodied shaped things here are the sites of excitatory synapses, and they're great because you can actually just count them, which makes them very handy. So it's a little bit of looking where the light is. And so what we asked very early on, I guess about 20 years ago, is there a sex difference in the numbers of these dendritic spines on the neurons in the male versus the female brain in the very early critical period for sex differentiation, which in the rat starts at embryonic 18 when they make testosterone and ends within a couple of days after birth, completely done by the first week of birth. So we just take the little pinkies and we dissected out their preoptic areas and asked, is there a sex difference? And obviously the answer is yes, or we'd all go home. So yes, males have about twice the density of dendritic spines. And this is actually what preoptic area neurons look like. They're very plain and simple. There's no dendritic tree. There's no arborization. But they are studded with dendritic spines. But the males have twice as many of those as the females. And so we're neuroendocrinologists, so we showed very early on that treating females with a masculinizing dose of estradiol increases the spine density to that of males. And I just have to pause for a minute and remind you that testosterone is the pro-hormone of estradiol, and in our rodent systems, estradiol is the masculinizing hormone. So everybody thinks estradiol, female, female, female. In this case, you kind of got to reverse it and think male, okay? Because developmentally, estradiol is masculinizing in our rodent animal models. So you say, okay, well, we're talking about spines, we're talking about synapses. This must be a mechanism that's going to involve neurotransmitters. That would be the obvious thing to think, and we weren't the first ones to think it. In fact, all of the effort at looking at sex differences, you could track the whole history of neurotransmission. First studies looked at acetylcholine, then adrenaline, oh, maybe it's serotonin, dopamine. Okay, none of those works. Well, how about oxytocin, vasopressin? No, they didn't work. Oh, maybe the neuropeptides, MPY, CCK, opioids, anything, GABA, glutamate, no neurotransmitter could explain the process of sex differentiation. You could mess it up really easily, but you could never substitute for the hormone. So we sort of said, it's complicated. It's reproduction. It's multifactorial. Nature doesn't play with fire. You're gonna have to have redundancy and et cetera, et cetera. But no, in fact, we were actually all wrong because we were all looking in the wrong place. And it was not a neurotransmitter at all. The whole process is actually initiated by a membrane-derived signaling molecule familiar to you all as prostaglandins. Prostaglandins, the ones that make us sick, give us a fever, give us aches and pains, and what we take are aspirin and Aleve and whatever your pain reliever of choice is for. So through a series of studies, a MD, PhD in the lab worked out a model system, Stuart Amato, that showed that testosterone aromatized estradiol binds to the estrogen receptor and leads to the transcription of the cyclooxygenase gene, which leads to production of prostaglandin E2. This acts on neighboring astrocytes to cause glutamate release in a calcium-dependent manner and cause the formation and stabilization of dendritic spines by activating the AMPA receptor. And so we got pretty far in this story about 10 years ago. And we confirmed that it was behaviorally significant by injecting female pups with prostaglandins directly into their brain during this critical period, right after they're born, just 2 days, we would inject right into their brains the day they were born, 1 day later, raise them to adulthood and ask, are you female, or are you male? And in fact, they would show that they had a masculinized brain. We gonadectomize them and give them male hormones to ask, are you male? And they were. And then likewise with the males, we would inhibit their cyclooxygenase gene, raise them to adulthood, and ask, are you male? And we found, in fact, they were completely asexual. They had lost all interest in sex. And no, the irony that we're using a COX inhibitor is not lost on me. So along the way on these studies, we found that a single injection of prostaglandin E2 on the day of birth, one single injection was sufficient to masculinize dendritic spine density and masculinize sex behavior. And of course, we kind of act like this was a very purposefully done experiment, but it was a graduate student doing the experiment. When you do development, you are a prisoner to when the rat decides to deliver. I know exactly what happens. She delivered on a Friday morning. He injected the pups on Friday afternoon, went out Friday night, Saturday morning. I think I'll do an experiment, see if one injection works. But sure enough, it did. It was just extraordinary, and even I just couldn't believe this. This can't be true. You can't just have one injection, one time, one exposure to prostaglandin. It's going to forever change the trajectory of something as important as reproductive behavior, right? It's all of evolution has brought to bear on that behavior. So we said we must be lighting a bonfire. We must be igniting a spark that is going to initiate another whole cascade of events. So there must be some kind of a positive feed-forward process that's going on. And when thinking about that, we turned our attention to another cell type, the microglia. Now, you guys are probably all with these, so this is the new cell du jour, right? Everybody's in love with microglia. We'd all ignored them for years, including me. I always just said, do they express GFAP? No? Okay, I don't care. I don't want to look at them. But we now are really excited about microglia. They are really fascinating. They're badly named. They're not glia, right? It's really unfortunate. They are, in fact, not even part of the nervous system. They are the primary immunocompetent cells of the brain, but they are modified macrophages. They actually are derived from the embryonic yolk sac, and then they migrate into the brain early in development, and the blood-brain barrier closes behind them, and then they take up residence in the brain. They've been mostly studied for their role in injury because they both respond to and produce prostaglandins. There's been a real revolution in microglia research in the last couple of years, and people are really completely rethinking them. We used to call them quiescent, and that they just sat there and did nothing and waited until there was an accident, and then they run to the scene of the accident and go, woo, okay, there's been an accident, and they clean up the mess, and they call in all of the other players, etc. But we now know they're actually doing a lot more than that. One of the things that they're doing is continuously surveying the environment. So they're tiled throughout the brain, just like astrocytes, so they all have a local micro-territory. And every microglia has a stable of neurons that it's responsible for. So the red here is the microglia, and you can see he's got his arms reaching out and embracing multiple neurons. And what they're doing is constantly checking on all of the neurons, saying, you okay, you okay, you okay, everybody's okay, okay, good, everybody's okay. Do it again, sound off, everybody okay, okay, okay. And they say that it takes about 2 hours for your entire brain to be surveyed by your surveying microglia. So they are turning out to be really fascinating, intimate partners with our neurons and glias. So the first thing we had to do was just ask, is there a sex difference in these microglia? Again, during this neonatal sensitive period, everything we do, unless I tell you otherwise, is right here, right when they're born. This is a coronal cross-section through the preoptic area. This is the anterior commissure. And all those little dark spidery things are microglia. They are IBA1 stained. And you can see how nicely tiled they are throughout the brain. And so Katie Lenz just went in and first asked, what's about number? What about density? Is there more than one sex versus the other? Turns out males have more microglia than females. And again, if we treat females with a masculinizing dose of estradiol, they increase their number of microglia to that of the males. This is within 2 days. This is all occurring very quickly. So it increases the number. And if we treat with minocycline, a tetracycline-based antibiotic that's known to inhibit microglia, we in fact prevent the ability of estradiol to increase the microglia number. So there's more, though, to them than just their number. They also come in all these different kinds of morphologies. They're incredibly plastic in the different shapes that they can take on. And their morphology is considered representative of their mood, of their state. And so an amoeboid microglia is considered activated. It's going to be producing more cytokines, more prostaglandins, and it's going to be, it's what you get after an injury or an accident. And the thin are the more ramified surveying microglia. They're considered the calmer type. So when we looked at the morphology, the sex difference was even bigger in that the males had many more of these sort of activated amoeboid-like microglia than the females. And again, if we treat with our hormones, we just rev up those microglia in the females so they become more activated. And again, this is also blocked by the minocycline, as you can see here, completely blocks the effect of estradiol. And this is what it actually looks like. These are the male microglia. They're kind of blebby. They're sort of angry, you know, a whole bunch of them. The females are nice and lacy and filigree. They're sitting quietly, behaving themselves, and waiting for tea time. So what's the effect of prostaglandins on these? Well, if we block the cyclooxygenase enzyme in males with indomethacin, which is kind of a pan-COX inhibitor, it's a little dirty, but it's very effective, we greatly reduce the activation in the males, and in fact, we bring him right down to the level of the female. But likewise, if we give the female prostaglandin, we actually wake up her microglia. So this is, in fact, our positive feed forward. So that was our spark that when we said it, we gave the prostaglandin, we transformed the microglia in the female brain, and then we went on to show that if she actually makes more prostaglandin in an ongoing process after we've activated her microglia. So how is it that we're getting more microglia in the male brain? Are there more microglia migrating in, or are there more being born? There are actually two phases of microglia migration into the brain. One, as I said, is very, very early on when it comes from the embryonic yolk sac. That would be way before there would be any sex differences in hormones. The other is more not as well understood, and it's around birth. And it's very hard to study migration processes because you have to close the blood-brain barrier, which is, I don't know any good way to do that. Anybody knows, they can tell me. So instead, what we did was focused on the proliferation question, and we used BRDU, bromodeoxyuridine, the thymidine analog, injected that around birth and the day later, and then just double stained with IBA1 to see if they were proliferating, and they are proliferating a lot. So what you're seeing is the green is the BRDU, and the red here is the IBA1. So like here's a microglia that's not proliferating, here's a cell that's proliferating that's not a microglia, but here is a recently born microglia, and here's one here, and here, and here, and here, and here. I think I have one more there. So they start expressing IBA1 within 24 hours of dividing. So they are replicating very quickly and differentiating very quickly, unlike neuronal and astrocytic precursors. And so when we actually count the number of BRDU cells, we can see that the males are making many more BRDU cells than many more IBA1 cells than females. So they have some kind of a baseline proliferative rate that's higher. They're presumably also dying at a higher rate in the male because otherwise his entire brain would be taken over by microglia. So that was sort of a lot of different kinds of disparate pieces of information. So I now want to I like to put things into cartoons. I think it helps get them across more easily and digestively. So this is our working model of how this process occurs, that we can go from prostaglandins and microglia to dendritic spines. So let's see if we get this to work. Oh, so just to orient you, we've got a neuron, an astrocyte, a microglia, and a neighboring dendrite. So we start with testosterone falling out of the sky, and it's going to be aromatized to estradiol, and estradiol is going to bind to the estrogen receptor, and it's going to transcribe the cyclooxygenase gene, which is going to convert arachidonic acid into PGE2, which is going to act on the neighboring dendrite at the EP2 and 4 receptors, which are cyclic AMP linked and protein kinase activated. An AMPA receptor is going to be phosphorylated to cluster at the membrane. Additional PGE2 is going to activate the microglia to make more PGE2 to act on those neighboring astrocytes and cause calcium-dependent glutamate release binding to the AMPA receptors, causing the formation and stabilization of dendritic spines. And I have really hard evidence for every step in that process but one, but I'm not going to tell you which one. Okay, so what are the surprises so far? First of all, is that prostaglandins, not neurotransmitters, are the primary target of hormone-induced masculinization. Second, microglia, non-neuronal cells, are a key partner in this process of sex differentiation. They have no relationship with neurons at all. And third is that early synaptic patterning that we've discovered actually endures into adulthood. I didn't show you the data, but we can discover it at birth, at 2 days of age, at 20 days of age, at 60 days of age, at 90 days of age. It is a organizationally permanently established sex difference. So to make that kind of graphically real, even though this is not what the dendrites look like in the preoptic area, if you look at the male, he's got these dendritic spines studded along his dendritic process, and the females are much, much sparser. but it's a density phenomenon. So it's like telephone poles along a wire, along the highway, right? And the male just has twice as many telephone poles as the female. So something is maintaining this pattern for the lifetime. And we all know that dendritic spines come and go. They're highly plastic, right? They're not telephone poles. They're actually very facile structures with a relatively short lifespan. So something is telling them to keep their distance. So what's the form of memory? Everybody's new favorite form of memory is we've rediscovered epigenetics, and this is how we imprint early life experiences via lifelong changes in gene expression. And of course, this concept originated in the 1940s with Waddington, and he created what he called the epigenetic landscape. And it's kind of the idea that your fate is determined, your cell fate is determined by epigenetic changes early in life, because every cell has your entire genome, right? But your different cells are expressing different types of different gene profiles. Two canonical types of epigenetics are the methylations of the DNA at the cytosines proximal to guanines, and the other is the histone modifications. Because this is a lifelong enduring change, we decided to focus on the DNA methylation changes as our potential source of memory. And of course, the DNA changes are brought about by DNA methyltransferases. And there are kind of two general classes of DNA methyltransferases, the DNMT1s, which are considered the maintenance methylation enzymes because they maintain the methylation pattern faithfully each time a cell divides, versus the DNMT3s, 3A and 3B, and they're considered the de novo methylation because they can respond to an environmental change and de novo methylate the DNA. And one of the things that happens when you methylate the DNA is that you often recruit methyl binding proteins that will then just block transcriptional apparatus from ever sitting down on the DNA, and by that way you sort of silence gene expression. This is the canonical view of DNA methylation expression of gene regulation, and anybody who's doing any epigenetic works knows I just way oversimplified it, and that there's all sorts of interesting exceptions that are appearing. But just starting with first principles, we just asked, is there a sex difference in the DNMTs and DNA methylation in the neonatal POA? And again, we're just focusing right again at this same developmental period. This is the thesis work of Bridget Nugent. And she did quantitative PCR of all three of the DNMTAs, as well as exhaustive Western blots. She did it on the day of birth, 2 days old, 4 days old, and 7 days old. She did 3 groups, males, females masculinized with estradiol. She had very large ends, and as you can see, she found bupkis. Absolutely no sex differences. And this is fairly unusual in our world to find lines that are so on top of each other. But not to be deterred, she said, well, there's not a different in amount, but maybe there's a different in activity. So she then turned her attention to a DNMT activity assay. It's a colorimetric and now fluorometric. It's just a 96-well plate assay. You put in a donor, you grind up your POA, and you put it in, and you compare males and females and see if there's any differences. And lo and behold, there was actually enormous differences in activity in the very young animals. So looking here on the day of birth, this is the normal female. There's a very high level of activity. Normal male, it's lower, and this is the female who was masculinized with estradiol literally just 6 hours earlier. So it was a very rapid effect. It persisted till day 2. By day 4, they had pretty much converged. And fascinating, by day 7, activity overall had greatly dropped. I had a hard time believing this data point and had them replicate it several times because it seemed strange. But in fact, it was true. There was a precipitous drop. And this is the end of the sensitive period for sex differentiation of the brain. Well, does this actually read out into differences in DNA methylation? She did another sort of antibody-based global DNA methylation assay just of the entire preoptic area. That's all we're looking at. We're just mushing up the preoptic area. And you can see females, percent methylation-wise, have almost twice as much as the males, and the females who've been masculinized with estradiol come down to the male level. At first you would look at this, and given what we just heard from Art, and we know about X inactivation, we say, well, of course females have more methylation. Every single cell has an inactivated X in it. But treating females with estradiol does not reactivate an inactive X, so there's something else going on. So this kind of faithfully replicates what we'd seen in terms of the activity assay, and we thought it was all well and good, and we sent it off for publication, very pleased with ourselves. And everybody believed it, but reviewer number 3. And, you know, it was just maddening because it was like, come on, it's all so clear and blah, blah. It was like, but we tried, we pushed, and like, no. And then finally the person said, look, what you're saying is really extraordinary. You're trying to tell us that the female brain in this region has twice the DNA methylation as the male, and extraordinary claims require extraordinary proof. Okay, fair enough. So we held our breath and made the big jump and went in and did whole genome-wide bisulfite sequencing. And we did the same 3 groups, males, females, and females masculinized with estradiol. There was this kind of approach generates so much data that it could not even be communicated to us electronically. It had to be mailed on a hard drive to be analyzed. And Bridget, by this time, this was her thesis research. She was already on her second postdoc, and fortunately had learned a little bit about how to do some bioinformatics, and she would access the University of Texas Austin supercomputer at night. She could get on for an 18-hour stretch, and it took 12 hours to just load the data from 2 groups to compare, and she would get 6 hours of analysis before she'd get kicked off and have to start the whole thing again the next night. So this is really not for the faint of heart, this kind of data. And all we could do is just skim the cream very much off the top. But what did we find? So if we looked at, this is a different way to present the data. This is the number of CPG sites that are methylated at 0 to 10% versus 90 to 100%. And this is how they've been out in terms of frequency. So if we look at those sites that are very highly methylated, the female is significantly higher than the female plus estradiol or the male. Interestingly, it shifts as we move down to 80 to 90%, and it then reverses again down at 0 to 10%. So it's almost like you've taken a mean and pushed it apart so that the females are at the extreme. If we look at those sites that are just 100% methylated, however, it exactly replicates what we saw with the global methylation assay. So those are the 3 animals in every group, and the females are about twice as high as the males and the females plus estradiol. So the note is that we think that global methylation assay is biased towards 100% methylated sites. Okay, so we were right, but not in exactly the way that we thought. In fact, this makes a lot more sort of biological sense. Where are these methylated sites? We just did an initial analysis. We would hope that they would be in promoters, but in fact, they're not very much in promoters or exons at all. Here's introns. The rest is all just in intergenic. Now, of course, most of the genome's methylation is in the intergenic region, but nonetheless, this is just where the sex differences are. And they also are not in CPG islands or even CPG shores. It's mostly other. So there's something going out in the deep, dark underbelly there of the genome that's very different in males and females. But the real question is this methylation important, right? Or is this just an epiphenomena? Is this really regulating the sex differentiation process? Well, the great thing about this reemergence in epigenetics is that there's lots of new tools coming on, most of them in the cancer biology world, and in particular trying to inhibit DNMT enzymes with DNMT inhibitors to reawaken tumor suppressor genes that have been repressed by methylation. So there's Zebularen, which is in clinical trials right now, and RG108. They act through slightly different mechanisms. One's a suicide inhibitor, one's a substrate mimic, et cetera. But they both block both the DNMT1 and DNMT3A, and they actually act as demethylases. Right, so they are DNMT inhibitors, but for reasons not fully understood, they do demethylate the genome. So Bridget went and just asked the simple question, does reducing DNA methylation by, again, injecting here the same times that we would have injected PGE2 or we would have injected estradiol or we would have injected minocycline, and then raising animals to adulthood, does that change their sex behavior and their dendritic spine patterning? So there's a full 60 to 70 days between our DNMT inhibitor treatments and our outputs. And first she looked at the synaptic profile, the dendritic spine. So here is the normal male. Here's a normal female. He's got about twice the number of dendritic spines as the female. Zebularen treatment doesn't do anything to him, but the female that got Zebularen 60 days later is indistinguishable from the normal males. And this is down here using, this was done with Golgi Cox. This was done with Western for spinophyllin. Here's the male versus female sex difference. Zebularen and RG108 both fully masculinize the dendritic profile in the female for a lifetime, because this is presumably pursued persisted since the treatment as a neonate. What about the behavior? When we study sex behavior, it's really nicely quantitative behavior. We can measure how frequently they mount, how frequently they show thrust-like behaviors, how long it takes them to show interest in sex. It's all in a very nice controlled environment. It's a much more pleasant way to do animal behavior, so it's why students like to come to my lab. They go sit in the dark, they watch animals have sex, they go home. So here's the normal sex difference. Males mount, females don't very much. Males with RG108 aren't affected, but females with RG108 think they're male. They're exactly like males. And this is here this is a male that we kind of uber masculinized with Zebularen. And I could pick all sorts of movie stars to say who he's like, but my favorite is Arnold Schwarzenegger. And then here's the normal sex difference, and here's the female with Zebularen. And then here's like, the thrust is much, much less frequent in females. Look at that sex difference is enormous, and the female with RG108 looks just like the males, likewise over here with the Zebularen. So in fact, we conclude that feminization, while it's still the default, is in fact the suppression of masculinization via DNA methylation. Right? And that masculinization is an escape, it's an emancipation from the depression of them from the female. Right? And I won't make any jokes about that because it's just way too easy. And it's estradiol that mediates this emancipation of the male by causing demethylation of his genome by activating inhibiting the DNMTs, or active, yeah. All right, so that was a surprise. We didn't expect that because, again, the female's just supposed to be the default and just supposed to be maintaining course, and the male is going to divert away. So, but then if this is about gene expression, what are the genes that are being affected by this neonatal DNMT inhibition? And to get at this, we have that data now in that genome-wide bisulfite sequencing, but actually the order of events was we did the RNA-seq first. We need to go back and mine that other data set, which we will at some point in the next century. But so we use RNA-seq to compare males, females with and without DNMT inhibition to see what would be the gene profile change. Here we did it in sort of real time, so we treated again on the day of birth and one day later, but then within 24 hours we did the RNA-seq so we could see what was happening at the time of sex differentiation. Now we thought we would have the age-old problem that there would be thousands of genes that would be different in males and females, and we'd say, what are we going to do with this mess? Now which one are we going to pick out? But using very low false discovery rates and a very low stringency for a sex difference, we found surprisingly few genes that were different in males and females. Only 7 genes were expressed at different levels. And again, surprisingly, we thought, of course, that it would be all biased towards one sex versus the other, but in fact it was almost evenly distributed. 34 were significantly higher in males, and 36 were significantly higher in females. So where among here can we get our masculinization genes? Well, if we look at the genes that were increased following DNMT in females, so these were the Zebularen-treated females. So they didn't have just 70 genes that changed. So this is female to Zebularen-treated females. They had over 300 genes, so consistent with the higher DNA methylation in the female. 300 genes were released. And in the male, we compare those to his 36, 25 of them were the same. So those are sort of our candidate masculinization genes. Here's a heat map of those genes. This shows this is the female treated with vehicle, male with vehicle, and the female with Zebularen. Here are all the gene names along here. Many of them were sort of unknown. A lot of them were just other transcription factors. There was sort of nothing really very exciting that stood out, with one exception, our old friend aromatase. We have known that aromatase is the key gene mediating sex differentiation for 50 years. Ta-da! We've made, what an advance we've just made. So it was both gratifying and incredibly disheartening that the only gene that we could really grab onto was aromatase, but it does seem to be regulated epigenetically. The great thing about RNA-seq, though, is that it detects isoforms. There's been very little attention paid to the notion of different splice variants or different promoter usage in males and females. And when we looked here, we found there were many more isoforms than genes that were differentially expressed in males versus females. 132 that were higher in males and 32 were exclusive, and then about 124 that were higher in females and 37 that were exclusive to females, which we didn't see with any of the genes. There was nothing that was exclusive. One of the things that did show up was spinophyllin, which is neurabin-2, which is interesting that that's our most consistent sex difference and that it's a splice variant. Looking at this heat map here is really quite striking. These are the normal control females, these are the control males, and these are the zebularen-treated females. So clearly this methylation is doing a lot to regulate the transcriptome in terms of promoter and isoform usage. Only about 25% of that difference is due to promoter usage. And this just kind of summarizes here the changes that we saw in the males, the 68 that were male biased. In the zebularen-treated females, 42 of them increased. And of the 32 that were male, only 16 now appeared in the female, about half. Of the 37 that were female, only 16 disappeared in the zebularen-treated females. And the 50 that were female biased, 24 of them were reduced in the female. Again, showing that methylation is doing more than just inhibiting gene expression. There could be gene networks, there could be lots of different things going on that are too complicated at this point. And it's interesting that in every case it was about half the genes that were regulated by the methylation. Okay, so now I've told you that aromatase is the only gene we found of any real interest, and so now you can say, well, wait a minute, what happened to the prostaglandins? What happened to the microglia kind of thing? Well, believe me, we looked very carefully for those genes, but we didn't see them. We did, however, do the ingenuity pathway analysis, and this is just comparing just males and females with no other manipulation. And when you look at what are the pathways, here's immune cell trafficking, cell-to-cell signaling, immunological disease, infectious disease. Down here we have inflammatory response, cell-to-cell, inflammatory response, immune cell trafficking, inflammatory response in the brain of perfectly healthy, normal males and females. So that was at least consistent with the idea that the prostaglandins were somewhere in the pathway. Well, when we did go back to the genes, we did find one surprise gene, and that was mast cell protease 2. Mast cell that can't be a coincidence. That's something, and I fortunately had gone to a graduate program called Institute of Animal Behavior with a woman named Ray Silver who had actually studied mast cells in the brains of birds many years ago. And as again, birds always so much inform us in the field of neuroendocrinology. So what are mast cells? As you all know, these are also non-neuronal cells. They are actually myeloid precursors from the bone marrow. They are found in every tissue throughout the body, including the brain. There's a resident population in the brain, but they also traffic in and out of the brain following injury, and they are central to allergic responses and first responders in injury. So they're both what give you a runny nose and what induce anaphylactic shock, right? And they cause rashes and they can kill you. So they are a very wily cell type. These are, of course, vesicles packed with signaling molecules, histamine, serotonin, GnRH. They make cytokines, they make prostaglandins. They are immune mediators. So Katie Lenz asked the simple question, is there a sex difference in mast cells in the neonatal POA? Looking again at this exact same time point as everything else. Great thing about mast cells, you don't even have to do immunohistochemistry. You just use toluene blue and stain for them, and they show up as these just little purple dots here. And so to count them is very challenging. Everybody in my lab is dying to do this experiment because you come and you go 1, 2, 3, 4, 5, 6. Let's go to lunch. Okay, I'm done. So there are very few. But as you can also see, though, if you know anything about mast cells in the brain, they're usually found out here. Like here's one down here in the meninges. These guys are all in the neural pill. All right, they are in the neural pill of the brain, of the preoptic area in particular. This isn't true everywhere. She quantified these across development, and on the day of birth, males have about twice as many mast cells as females. Interestingly, males show a huge peak of mast cells within the first week, and then they decline. They go down to very, very, very low, and then there's a slight uptick again in adulthood, but the sex difference is gone. Is this a hormonally mediated sex difference? This again just shows you an overall mean on PN4, males more than females. Treat females with estradiol, and they make more mast cells. We looked to see if they are proliferating. We could not find any evidence of that, so we think this might actually be a chemoattractant type phenomenon. So mast cells degranulate to dump their contents. They are the first responders at an accident, right? So when there is a brain injury, they rush to the scene of the accident, dump their contents, woo, and call in that calls in the microglia and everything, and then they clean up the mess. So because they're so important, there's lots of tools to study them, both to cause them to degranulate with chromalin, or you can prevent them from degranulating, with chromalin, or you can induce them to degranulate as a tool, because there's a lot of interest sometimes in getting both responses out of them. So what Katie did was the same paradigm we've done all along. She injected the animals either with chromalin or 480, 4880, excuse me, and then just asked what does that do to the neurons of the developing brain. So she did this experiment here. It was actually done in culture with preoptic area neurons in culture, and you can see here the dendritic spines on these neurons. Look at this one, it's just studded with dendritic spines in culture. So here's the male, and here's the male who was given a mast cell stabilizer. It didn't have any effect. Female sex difference here, but if we gave the female conditioned media from stimulated mast cells, this is her, she just grew dendritic spines like crazy, and if we gave her the stimulator with no microglia around, there was no effect. So there's no mast cells here, this is a mast cell conditioned medium, but there have to be microglia. So the question is, does mast cell degranulation impact microglia morphology? And this then was done in vivo. And here she has the normal sex difference. This is an activated microglia. Here's the male versus the female. He's got many, many more activated microglia. If we calm down his mast cells and tell them to stop degranulating, it calms down his microglia. This means that his mast cells are constitutively degranulating all the time. And if we stimulate her mast cells, even though she has many, many fewer, it's enough to rile up all of her microglia. And you can see that quite here, clearly here. Here's that male and female you saw before. Here's the female with degranulated mast cells. Look at all those blebby, just riled up, beer drinking, football watching microglia versus the male's lovely, hands crossed on their lap, quietly waiting for instructions and surveying their surrounding. Okay, so what does it do to behavior? I know a person who studies sex differences should not put all those biases in there. I know, that's terrible. But I can't resist. Okay, so what about behavior? This is looking at the number of mounts, right? And here's the normal male and here's the normal female. They don't show very many mounts. The male with the stabilizer wasn't affected, but the female treated with the stimulator started to behave like a male. We actually do sex behavior over 3 weeks because it's actually a learning process. Males get better, and so you can see even normal females will start to behave like males if they're given testosterone for a long time. But this is the female who was given the stimulator. She was even better than the male vehicle on the first test, kind of thing. So they were truly masculinized in their behavior just by stimulating their mast cells. So as we said, mast cells are involved in allergic responses. So what happens if a pregnant dam has an allergic attack? So what Katie did was she took virgin females and she made them allergic to egg white, egg albumin. And while they were pregnant, on gonadal day 15, she caused an allergic reaction by intranasal egg white. And then we looked at their pups at PN4 and looked at their mast cells, and then we looked at their sex behavior in adulthood. So vehicle is a normal pregnancy, OVA is an allergic pregnancy, and this is looking at the number of mast cells, no big effect in the males, but in the females, a huge increase in the number of mast cells in the PN4 fetuses. They were also, there was a big increase in degranulation, which I'm showing here. In both males and females, the mast cells were more degranulated. But more importantly, what about the behavior? So this is the normal male behavior. They were perfectly normally masculinized. Here's the sex difference. Normal females do not show much mounting behavior, but the females that were in a pregnant dam when she had an allergic attack showed male-like sex behavior. So to come back to our original model, there are at least 4 cell types involved in the POA masculinization, and 2 of them are non-neuronal in origin, the mast cell and the microglia. And that was also a surprise. And then this is just a little kind of side food for thought, controversial idea. When you think about the default sex is the female, one would predict that female behavior would be less variable, like in humans, right? So females should have a more consistent behavioral gender identity phenotype behavioral pattern. And males, because they are the divergent sex, or the differentiated sex, they should vary along a big continuum. But in fact, in the human sex research, it's exactly the opposite. Females show much greater variability than males. So could this be a naturally occurring source of variation in female behavior? Just food for thought. Okay, so that's my story in the preoptic area, but that's really limited to sex behavior, because that's what the preoptic area does. And you want to know, does any of this generalize to other brain regions that are more relevant to things like neuropsychiatric disorders and their gender biases? And also, the NIH has told me they won't give me money to study sex anymore, so damn. So we've had to move on to other brain regions. So we, of course, if we can't do sex, we'll go to play. And so we've been studying play behavior in the amygdala because the amygdala controls juvenile social play behavior. And we discovered years ago a sex difference in neurogenesis in the developing amygdala, such that females make more new cells than males. So we just did a classic BRDU quantification of the number of cells and looked at as they were growing up. And it was just a straightforward neurogenesis study, or cell genesis. And this sex difference here is not particularly robust in this experiment, but often it's about twice as high, females making more new cells as males. And the big question is, how is this happening? First question is it prostaglandins? And the answer was no, but the surprising answer was it is another membrane-derived signaling molecule, endocannabinoids. Endocannabinoids are also derived from arachidonic acid. And what we found was that newborn males have a higher endocannabinoid tone in the amygdala than females. So they have more anandamide and more 2-AG, and they have less of the degradative enzymes, fatty acid amide hydrolase and MAG lipase. And just to show you the way that endocannabinoids usually work is they're kind of presynaptic inhibitors of glutamate release, but we're finding that they do a lot more than anybody thought. So we know that females are making more new cells in the developing amygdala than males. So what is the impact of endocannabinoids on that? If we treat females with either a CB1 or a CB2 agonist, we actually reduce their level of cell genesis down to below that of males. We have hit males with a 100-fold range of endocannabinoid doses, and we can't budge his cell genesis. They're just, they're just naturally high, and so we just can't get them any more responsive. So they're completely unresponsive to the endocannabinoids. Okay, so you'd think we would start wanting to look at cell cycle and things like that, and how are the endocannabinoids regulating this proliferation. But along the way, we looked back at those microglia again, and a new graduate student came into the lab, Jonathan Van Rissen, and he started looking at the microglia, and he said, what are these funny cup-like things all over the microglia? And Katie Lenz had said, oh, I saw those. I didn't know what they were, so I just ignored them. Turns out these are really important. These are phagocytic cups, and that's a lot of what microglia do, and particularly when they're in the surveying state. You think of the amoeboid microglia, they come and cart away the dead bodies after an accident, but these surveying microglia are known to reach out, and when a cell's in trouble, they engulf it and pull it in and devour it. Right? And that's what you're seeing right here. Here's the cell body of a surveying microglia, and here's its long arm of the law reaching out and engulfing this cell that it seems to be unhappy with. Here's some other images of phagocytic microglia. They can have these large compound cups. This is our Mickey Mouse microglia. Here's a double B cup microglia. Here's our lacrosse microglia. We're in Baltimore. But it's very common that they have these very long processes, and these are huge phagocytic cups, right? These guys aren't nibbling. These are very large in size. And so when we quantified them, we found that the males actually had many more of these phagocytic cups than the females, only in the medial amygdala, okay, and only during this developmental time point. And if we treat with the agonist, the CB1 is significant, CB2 is just trending, the females increase their phagocytic activity of their microglia. So what are they eating? Really, the only candidate is other cells, and it is definitely other cells. The DAPI is staining nuclear material. This one here has also CD68 to show it's truly lysosomal activity, that they are digesting what they're engulfing. You can see here, I don't know with the lights on, but you can see there's actually two. This is a two-fisted drinker here. He's got two cells over here, and another, this is from the same cell. This one down here has multiple cells inside the cup and over here. And it's known now in microglia that they'll kind of go on a feeding frenzy, and they'll engulf a whole lot of cells, and then they just stop, and they just have to kind of digest everything. What we don't know is if then they go back and feed again, or if they just die and get washed out of the system. It's, are they mayflies, or do they exist longer? So the big question here is, are the microglia eating the newborn cells in the males? So to come back to the original sex difference, this is more robust than what we usually see in the male and female. We treat the female with testosterone, and she reduces her neurogenesis. But more importantly, if we treat with minocycline to block that phagocytosis during this period, all of a sudden the cells appear in the male. And it turns out that the sex difference in cell genesis is secondary to endocannabinoid-induced myoglial phagocytosis. So it's probably not a cell birth difference as much as it is a cell death, perhaps cell murder difference by the microglia. So that was a surprise. So my big picture conclusions are that the cellular mechanisms establishing sex differences in the brain are unique to each endpoint and region. So I just talked to you about the POA and the amygdala. We could go on and on about the ventral medial nucleus and the hippocampus and the PVNST and et cetera. They all seem to have unique mechanisms. There's no one unified field theory for sex differences in the brain. And then second is that variables other than classic regulators of neurodevelopment and plasticity are the regulators of many sex differences. And this sort of leads me to two competing, but I think compatible concepts. First is that there's no such thing as a male brain versus a female brain, despite covers of Scientific American or Time magazine or whatever, and there's no blue brain, no pink brain, or anything like that. Because we know that the mechanisms are unique in every different brain region, by definition, we're dealing with a mosaic. Right? Because there's no way that there's nothing that's unifying the process in males and females. Unlike your gonads, which are going to go all male, or all female, your brain is really a heterogeneous organ, and so we're going to wind up with individuals along a variance of female brains with very little male components, or female brains with lots of male components, male brains with lots of female components, and vice versa. And that's an important thing about, it's a great way to increase diversity and stuff, but it's also important about not being predictive of any individual's behavior. But a mosaic is not a blend, right? So I very carefully avoided the word continuum. There's not a continuum of maleness and femaleness. There is a dichotomy, because after having done this for 25 years now, and every single anatomical or neurochemical or biochemical endpoint we look at, we see the same pattern and magnitude of sex difference over and over and over again. It's almost always twice as much in one sex versus the other. I've got male versus female here, but it could easily be flipped the other way. Over and over again, it's twice as much. We give the female hormone, and we bring her right up to the male level. We almost never supersede the normal masculinization. We rarely make an Arnold Schwarzenegger. They're really the rare exception. And if we give the male more hormone, he doesn't become more masculinized. The other important point is that the variance is also really small and highly consistent, which is not what you would see if you had a continuum, right? You would have a lot of variance. You would have small magnitude differences with big variances, and instead we see high magnitude differences with small variances. So it's almost like something is keeping down the variability. But something is also pushing the sexes apart. So when we return to Waddington's epigenetic landscape, he also proposed the concept of canalization, which has many, many applications, but is particularly used by evolutionary biologists as a way to explain species integrity, right? And there are clear cellular mechanisms for canalization so that species don't respond to every little insult in the environment and go off mutating themselves into new species. They have to stay where they are. But canalization has never been, and the idea is you go down one canal and you have to stay there, it's never been applied to the concept of sex differences. So could sex differences in the brain be canalized so that you go down one path or the other and you can vary within that path, but only so much? So something's going to keep the two sexes apart, but something's also going to keep them together. And there's obviously important evolutionary reasons why you do not want the sexes diverging. And it's a lot like what Art was talking about compensation, too, just for health and disease, but also for purposes of reproductive fitness. We have to make sure that they stay close enough together. And what are potential agents of canalization? I mean, in humans, it could be environment and experience. Stop playing with that doll, get back to your trucks, those kinds of things. Hormones, epigenetics, microRNAs are clear agents of canalizations, chaperone proteins, and even transposons have been all proposed as agents of canalization. So why might this be interesting? Just big picture idea, being male, as we've been learning about all day, is the leading biological predictor of your relative risk for developmental neuropsychiatric disorders such as autism, early onset schizophrenia, but also neurological disorders like dyslexia, attention deficit, stuttering, Tourette's, all kind of disorders of wiring. And also early life inflammation, gestation or early childhood, is the leading environmental predictor of your relative risk of a developmental neuropsychiatric disorder. So I'm leaving out genes, right? So I'm bringing up hormones and environment as the two variables, and they have to be acting on a genetic background. But the question is, are males closer to a vulnerability threshold with their higher rates of prostaglandins, more microglia, more mast cells, more inflammatory cytokines, et cetera, so that when an inflammatory insult does occur, are they just, standing that much closer to the cliff, and they're going to go right over the edge, as we all know little boys always do. Get away from that edge, you're going to die. Okay. And could canalization contribute to gender biases in disease, such that along the male canal, are we somehow, in some biological way, closer to autism, ADHD, dyslexia, and schizophrenia, and versus the female side, we're closer to depression, anorexia, OCD, anxiety, etc. That's going to take a lot of work to get to that at a biological level, but it's just a food for thought idea. And then I will end by thanking my funding agencies. And then also, I have been very fortunate to have an excellent continuous cast of great graduate students and postdocs over the years. And I thank you for your attention.